Welcome everyone to our 2020 Elakha Alliance Virtual Symposium. This is presentation number 7 and the final presentation around the topic of sea otters and Oregon's kelp ecosystem. Although, as we heard, it's also about our eelgrass and estuarine ecosystems too. We want to thank our co-sponsors, the Hatfield Marine Science Center and the Oregon chapter of the Wildlife Society, for their help in getting the word out and informing a great range of people. We've had super attendance, and I really appreciate everybody coming to learn as we are learning. And thanks to the Meyer Memorial Trust for financial support for the Elakha Alliance. And I encourage you to go to our website for lots of different reasons. You can go to podcasts that feature Tim Tinker, Jim Estes, Sarah Hamilton, Scott Groth, a whole bunch of other folks on these topics in depth. You can link to a lot of our different learning resources, and importantly, you can also help to donate to our effort here. A warning to everyone again, for those new to this session, we will be sending out a questionnaire to you, or a link to a questionnaire, and asking you to fill that out, and that will help us for the next time around. So you are on mute, and if you're not, please mute yourself. And if you have questions for the presenter, you may send those in the chat room or Q&A to John Goodell. And we're going to go for about 55 minutes, but we're going to let this go a little longer if we need to. And if we like, we can circle back at the end and bring Brent Hughes in to answer some of the questions we were not able to get to previously. So I'm really happy today to have this gentleman, although he might not admit it, this gentleman on our panel. Tim is one of those people that pops up everywhere in the literature as either the lead author or co-author. And so his reach across the world of ecosystems and sea otters is vast. He's widely respected, widely published senior marine ecologist. He's a principal research ecologist with Enhydra Ecological Consulting, and back in his home turf of Halifax, Nova Scotia. We're back there. It's dinner time for him. He currently holds appointments at several universities in the US and Canada, including the University of California at Santa Cruz, where he spent many years. If you look up the literature on sea otter related research, he shows up as a contributing or co-author on a very high percentage of papers. He has mentored and advised literally hundreds of graduate students who now populate the sea otter and ecological related research world. He has frequently participated as an expert advisor to the U.S. Marine Mammal Commission, the U.S. Fish and Wildlife Service, the Canadian Department of Fisheries and Oceans. He has co-authored the ESA recovery plan for the Southwest Alaska sea otter and contributed to workshops convened to develop population viability models for marine mammals and to investigate ecological impacts of killer whale predation in the North Pacific. And when he finds the time, Dr. Tinker is the lead author on a feasibility study for the Elakha Alliance that will assess the issues and factors to be considered in restoring a population of sea otters to the Oregon coast. We have asked him to provide an overview of what such a study entails and the key scientific considerations that will need to be addressed. So it's my pleasure to welcome Dr. Tim Tinker. And Tim, you may share your screen now.
Okay, thank you very much, Bob. Let's see here. Actually, let's start video so you can see that I actually don't look quite as young as I did in that picture. Yes, and let's think screen one.
There you go. There you go.
All right. Well, thank you very, very much, and for that very generous introduction. I think my role these days in the COVID world is primarily helping my daughter with her grade 8 math homework, but I still try to find time to do some sea otter related things tucked in around that. So thank you very much for having me here today, or remotely here today, to talk a little bit about some of the topics that I've been very interested in for a long time now, regarding ecological, demographic, and geographic considerations for for sea otters in general, and in particular as they may be relevant when thinking about potential recolonization of the Oregon coast. And Bob and John sort of gave me wide range to kind of address whatever topics I thought might be interesting in this context, which was perhaps dangerous. So I'm not sure if I'm going to be covering exactly what they thought I was going to be covering or not, but hopefully we'll cover some interesting ground and maybe see something, learn some things that might be useful considerations as we think about sea otters in Oregon. So sea otter ecology and demography. Why are these subjects important, or at least important to anyone else other than me? Well, I'm going to be able to convince you that they're important because they may help us address some of the questions that we may have about potential ecosystem effects and also socioeconomic impacts of sea otters should they return to Oregon. And perhaps one might say when they return, because as the population spread, eventually at some point they are going to, in all likelihood, return to Oregon, either from Washington or from California. But they may, through human interventions, they might arrive there sooner or later. So it is my feeling that some of the recent findings in particular that some of which you've already heard about from other speakers about sea otter ecology and population biology can really help us think about and even anticipate the nature of some of these ecosystems effects and socioeconomic impacts of sea otters. And even more specifically about their likely timing and geographic patterns, depending on when and where sea otters arrive back in Oregon. So that's sort of what our ultimate goal is. But to get to that goal, we're going to cover quite a bit of ground today. So I'm going to move along. I'm sure you'll probably have some questions about some of the things I'm going to cover, because I am going to go pretty rapidly, but we can talk about those things in the question and answer. So I'm going to start with a very quick basic ecology overview of sea otters. And I'll point out at the top of the screen here, you see in blue text a reference here. This is to a chapter in the Encyclopedia of Marine Mammals. And throughout this talk, there will be several times there'll be a reference like this, a citation to a paper or a book chapter will show up. And I think we'll be able to put all of those as reference documents somewhere for you to download. I think. I'm not 100% sure on that, but that's what those are for. So, for more information on sort of basic ecology of sea otters, you can refer to that chapter or many other references. So sea otters, as most of you know, are a mustelid carnivore. That makes them a little bit different from most other marine mammals, which are pinnipeds or cetaceans. Sea otters have the status of being the smallest completely marine mammal, although there are some other smaller otters who spend a good portion of their time in marine environments, as you just heard about in Brent's talk. But in terms of mammals that spend their entire lives in a marine environment, sea otters are the smallest. And that has relevance for several reasons that we'll talk about. Their distribution is limited to coastal marine habitats around the North Pacific, from Hokkaido, which is the North Island of Japan, around sort of the Russian coastal areas, Kamchatka Peninsula, and then the Commander Islands, right around the Aleutian Islands, and then down the west coast of North America. All of these areas, they're limited to this very narrow coastal fringe, and I've said here less than 100 meters of depth, because that's about their deepest dive depths. But in fact, the vast majority of their diving is in 40 meters or less of water. So really, it's this extremely nearshore fringe of coastal habitats, the extreme nearshore marine habitats. Their diet is, in most areas, completely consists of subtidal and intertidal benthic invertebrate species, a wide range of different benthic invertebrates, definitely well over 100 species in many areas that we've studied them. They do eat actually fish in a few areas, particularly in the Russian waters, are definitely in the Russian Commander Islands, and then in some areas of the Aleutian Islands. Their diet can consist up to about 8 to 10% fish. Species, things like kelp greenling, and some flatfish. But in most of the rest of the areas that we've studied them, they don't consume fish at all. They have relatively small home ranges and relatively limited dispersal as compared to other marine mammals. So if you're used to pinnipeds or cetaceans that sometimes range over hundreds of kilometers or entire ocean basins, sea otters are quite a bit different. Individual animals spend their lives in relatively small areas of the coast, with individuals' home ranges, lifetime home ranges, often being only 5 to 15 kilometers of coastline. Their average lifespan is 14 to 18 years. Females can occasionally live longer, up into the 20s, but usually it's that range. And in terms of reproduction, they have a polygynous mating system. Some adult males are able to defend small coastal territories wherein they maintain exclusive access to females that reproductive age females that occur in those territories. Reproduction in sea otters can occur throughout the year. They can mate throughout the year and they can produce pups throughout the year, although there tends to be some seasonality, particularly as you move farther north. So in northern populations, most pupping occurs in the spring, but although in California, there's both a spring peak of pups and then a fall peak of pups. And then some pups can be born at any time during the year. For females, their age of first reproduction is about 3 years of age, and after that age of first reproduction, they're going to produce 1 pup a year for the rest of their lives. They're female sea otters, once they reach reproductive age, are always either carrying a pup, so they have a pup dependent upon them, and that lasts about 6 months if it's successful. Right after weaning, they come into estrus, they mate, and then they begin a 6 months gestation period in preparation for their next pup. So it's a hard life as a female sea otter. So sea otter foraging ecology, we're going to spend a little bit of time on this for fairly obvious reasons, I guess. Sea otters, as you've heard in previous talks, have these enormous ecosystem effects. And the reason they have those effects is because of what they eat and how they eat it. So understanding sea otter foraging ecology is kind of key to understanding how they're going to affect food webs, including species that we may be particularly interested in, such as commercial fisheries. So sea otters, I mentioned their small size. They also are one of the only marine mammal species that do not use blubber as a thermoregulatory device. They have no blubber. In fact, they have almost no body fat. Their way of keeping warm is to have the thickest fur of any mammal and to have one of the highest mass-specific metabolic rates of any mammal. So they spend their lives in this sort of thermally challenged state with very high elevated metabolic rate, and a high metabolic rate means they have very high energy demands. So they have an extremely high rate of food intake. About 25% of their body mass every day has to be consumed in terms in benthic invertebrates. And it is in fact that aspect that makes them a keystone species. That's why they have such profound effects in marine food webs. So understanding sea otter diets and foraging ecology is therefore critical, as mentioned, for anticipating their effects on ecosystems and on people's livelihoods. There are, though, a number of complexities when we're studying diets. It would be nice if we could just sort of describe a sea otter diet, and that would be that. However, the diets are not fixed. They vary quite a bit. They vary as a function of habitat, and you just heard in Brent's talk how some of their food web interactions in salt marshes and estuarine eelgrass beds are quite different than they are on the outer coast, and they therefore have different diets and different food web effects. So habitat is one of the things. Different habitats predict different diets. I'm not going to talk too much more about that. I'm going to mostly focus on outer coast sea otter ecology for the remainder of this talk. Population status is another factor that affects their diet, and we're going to talk quite a bit about that, about how their diets change as population density changes. And finally, there are individual differences in diet, which we've spent quite a bit of time studying and learning about, and those also turn out to have really important consequences for food web dynamics, as we'll discuss. So there are all these complexities. Luckily for us, sea otter diets are one of the easiest diets to study of any large carnivore species. And that has to do with their sort of unique behavior and where they live and how they feed. So as mentioned, sea otters are in this very nearshore fringe of marine habitats. It actually means that almost all their feeding occurs within sight of land, particularly if you have a high-powered telescope, such as Michelle has here in this picture. So if you're armed with a really nice, powerful, high-quality field telescope, you can see, even an otter half a kilometer or a kilometer away on the coast, you can pretty much see exactly what they're eating. That's because although they capture their invertebrates in the bottom of the ocean, they bring all their food up to the surface to consume, and then they do that by lying on their backs and even putting their prey on their chest like a tabletop, and then eating the prey one species at a time. So that means we can sit there with these telescopes, or actually stand there and get a really bad neck ache over hours of time. And we can record every item that this otter captures from the bottom and puts into its mouth. So what data are we collecting these observations? Quite a bit. We're collecting, these are the basic sort of types of information that we record from feeding sea otters. And we've recorded feeding data from sea otters everywhere that they occur in the North Pacific, from the Russian Commander Islands and Kuril Islands, right around all the populations in the West Coast. So we're recording the duration of their dives, both the time underwater, retrieving prey, and the time at the surface, handling the prey, which prey species they're eating, what size, and we can measure the size because we know in great detail their paw widths, because we've captured thousands of otters and measured their paw widths. So their paws are like little rulers that they're holding up with each prey item. We record the number of items per dive, handling time, whether they shared prey with a pup or whether prey was stolen by another otter, and then a variety of other things. And we take all that information and we can analyze it in using sort of a not too complex model, but a model that accounts for some of the specifics of sea otter foraging and some of the biases inherent. So we calculate caloric content of each captured prey item by species and size class. I'm going to show you how we know that in a second. We account for capture and handling time and success rates. We account for the prey shared with pups or stolen by other otters. And then we account for some of these measurement biases and uncertainties associated with certain smaller prey items, for instance, are harder to see and are more likely to go unrecorded, and that sort of thing. But we're able to account for those in fairly clever ways. We've measured all the different prey types that they eat. We've gone out, we've collected those prey, we've developed size biomass curves and measured their energy content using bomb calorimetry. So that, for instance, with this mussel here, we can compare it to her paw width. We know it's 1.2 paw widths in size, and we can therefore, from these functions, figure out exactly how many grams of edible biomass and how many calories she's consuming. We put all that together and we end up with a measurement of the energy intake rate, so kilocalories per minute of foraging time, and then diet composition in terms of the proportion of consumed biomass. And we have done this, as mentioned, in populations all around the North Pacific. Here are some of the areas that we've collected sea otter data, and you'll see we've got these little speedometers for all these different populations, and or thermometers, however you want to think of those. And what those thermometers are showing are the rates of energy intake in different populations, with the maximum being 20 kilocalories per minute of foraging time. So somewhere like Glacier Bay, when they first colonized Glacier Bay, that they were up there at almost the maximum rate of energy gain, although over time that rate of energy gain declined. When we put all that together, we notice some pretty consistent patterns. And the most striking pattern that we notice is that in populations that have been recently established, where sea otters are still at relatively low densities and they haven't been there for very long, they tend to have very high rates of energy gain, usually about 15 to 20 kilocalories per minute. And then over time, when they've been in areas for longer periods of time and they've reached higher densities, we see that rate of energy gain drop, and ultimately reaching the sort of rate about between 5 and 10 kilocalories per minute. So the rate of energy gain is more than twice as high in recently established populations as it is in these high density, long established populations. So that's I've mentioned before how diet and foraging success are related to population status. And that's when I say population status, that's what I mean. Status of population with respect to its density or its potential maximum density, which is something we call carrying capacity. So in recently colonized areas, we have this high rate of energy gain. And then as populations grow, they tend to deplete their most preferred prey item. And their most preferred prey are what they feed on most heavily when they first arrive in an area. These are the large energy-rich species. But as those large energy-rich species decrease, and also as sea otters become more abundant, so per capita abundance is going down, they have reduced rates of intake, and then they begin to broaden their diets. And the prediction from optimal foraging theory, sort of a whole branch of theory to do with how animals forage, is that we expect predators to expand their dietary niche as competition increases within a population, and their preferred prey become depleted. They begin to add more and more unpreferred prey into their diet. So this means we expect to see greater diet diversity in high density populations where food abundance is reduced. So we've gone out and we've tested this hypothesis. As sort of the basic thing that scientists do. We have a hypothesis, we go out, we set up studies, we collect data, and we see how our hypotheses measure up against nature. So we've measured these both rates of energy gain and also diet diversity in populations of sea otters in different areas. And this one study we did in California, we took two very similar areas that had almost identical compositions of rocky benthic shorelines, subtidal habitats. One was Cambria on the central coast of California, and the other is San Nicolas Island. And in one area, San Nicolas Island, otters had only been for a very relatively short amount of time, and the population density was very low. There were only about, at that time, actually about 25 otters on the entire island. Whereas in Cambria, they'd been for many, many years, decades, and were at very high population densities. And sure enough, the predictions pretty much matched what we expected. So in San Nicolas Island, they had a very high rate of energy gain, about 15 kilocalories per minute, and not a very diverse diet. The diet was dominated mostly by large red urchins, but then kelp crabs and then these large marine snails also were the two next most abundant prey. So this H metric measures diet diversity. It's called the Shannon-Wiener Index, and it was about 1. When we went and looked at otters in similar habitats in Cambria, what we found was quite different. The rate of energy gain was less than half of what it was in San Nicolas. It was about 6 kilocalories per minute, and the diet diversity was much higher. So there you can see this sort of histogram showing all the different species that otters are consuming at the population level. So that matched pretty much what we expected. However, we did another thing. We started, because in both these studies, we actually had gone out and tagged individual animals. And both these studies lasted about 3 years, which is how long the radio tags that we equipped these otters with last. Oh, I apologize for that. I'm going to turn off this phone. So we had tagged these individual otters, and we were able to follow them over a 3-year period while they had their active radios. And what we discovered is in San Nicolas Island, all the individuals had relatively similar diets. In all cases, for every tagged individual, and I've just shown 3 random adult female animals, the urchins make up the highest portion of their diet. But when we looked at individual diets, and these are again 3 similar aged females from Cambria area, San Simeon or Cambria, and they all had overlapping home ranges. In fact, their home ranges were almost identical, and yet their diets were completely different. One specialized on small brown turban snails, another one specialized on cancer crabs and abalone, and the third one specialized on clams and fat innkeeper worms. And there were other females that had different diet specializations. So this was one of the first studies that was done of a large carnivore where we were able to look at individual diets over many years, and subsequently followed otters over their entire lifetimes, and discovered that they maintained these specializations for their entire adult lives. Since then, a lot of other people have looked at a lot of other species and found that this type of individual level specialization within a population is not rare at all. Actually, ours was one of the first reports of this, but since then there's been hundreds. So it's starting to look like this is a relatively common phenomenon in a lot of animal species, which is very interesting. What we find in high density populations in California is that there are 6 diet specialist types that we find in almost every area of similar habitat. This is on the outer coast. We find different patterns in estuaries, which I'm not going to talk about today, but those are also interesting. But along the outer coast, these are the 6 most common diet specialist types. And interestingly, we find them in about similar ratios wherever we look in the outer coast. And there's a bunch of papers that we've written talking about this. So here are some of them here, and we'll try and make sure that these are available for you to download. But this has been really an interesting journey. We've used different methods as well as observation. We use stable isotopes to try to understand the patterns of specialization, what causes it, when it's likely to occur, and how the implications for sea otter health. One of the things that this diet specialization does is complicate food web responses. And a great example of that actually is provided by the Monterey urchin boom that you heard about if you tuned into Josh's talk yesterday evening. I think that was yesterday. You heard about a really interesting phenomenon that's occurring in Central California around Monterey after 2014, when we had this explosion in purple urchin abundance, something that had not been seen ever since sea otters had arrived in the Monterey area about 40 or 50 years earlier. And that was, we're still actually in the midst of conducting that study to learn about some of the patterns, and Josh told you some really interesting trends and findings that we're having in terms of the implications of sea otters feeding on urchins along the edges of these urchin barrens, and the potential resilience that confers to kelp forests. And one of these graphs Josh showed you in the talk actually, and that's how the proportion of sea otter diets consisting of urchins increased after this urchin boom. Prior to the urchin boom, urchins made up about 10% of sea otter diets in Monterey, and after the urchin booms, that more or less doubled to about 20% of sea otter diets. However, Josh did not mention, maybe because he thought he'd be kind and leave the fun for me, but in his study, we were actually able to look at what individual otter diets were, because we tagged a bunch of these otters in that study. So we're actually able to see how we can break down that change in urchin diets to urchin specialists, that is, otters that were specializing urchins prior to the urchin boom, and non-urchin specialists, and that is otters that specialized in other prey prior to the urchin boom. And what we see is that for urchin specialists, there's really no change before or after this urchin boom in the proportion of their diets that made up of urchins. In both cases, their diets were about, on average, 40% urchins, and then with the remainder consisting of other prey. For non-urchin specialists, there was this an increase, although it was not as great as we actually expected. So urchins, for non-urchin specialists, urchins made up much less than 10% of their diet, and that increased to about a little over 10% of their diet after the urchin boom. So if you sort of run the math for these two groups of animals, it actually does not add up to the pattern that you see here in terms of the population level response. And the reason it doesn't is because what we haven't controlled for here is the proportion of the population made up of urchin specialists. And it turns out that's what really changed. So prior to the urchin boom, this was the proportion of urchin specialists. And after the urchin boom, it went up to about a quarter of the population specializing in urchins. And most of these animals were actually animals that were juveniles or subadults at the beginning of the urchin boom, and basically came up through the University of Abundant Urchins and learned to be urchin specialists because that was the most abundant prey. So most of the older animals that we had tagged and studied retained their previous specializations. They didn't change. One of our older tagged animals did, so she made the switch, but others did not. And so most of this increase is represented by younger animals that have sort of come up in this world of abundant urchins. So it turns out that is how the response happens, is by an increase in the frequency of urchin specialists. This is something, now I mentioned to you before, the specialization really only occurs in dense populations like that at Monterey, where the population has been established for many years. So that means that this response to the urchin boom is fundamentally different than it would be for a colonizing population where no animals are specializing. They're all eating the most abundant prey, which is urchins. And that's why we think some of the differences that we're seeing in Monterey between when urchins have become abundant and how otters differ in the response to this sort of suddenly super abundant prey. We think those differences are the differences associated with a superabundant urchin availability in a population that was already near carrying capacity and where diet specialization was firmly established, as compared to an expanding population where individuals are not specializing. So this individual specialization does have implications for how sea otter populations are able to respond to variation in prey. Okay, so to summarize, sea otter populations grow in abundance. This leads to reductions in key prey, lower energy intake, diet diversification at the population level, which appears to be driven mostly by individual specialization. As they're having lower quality diets, that means they're in poor nutritional status, poor body condition. This leads ultimately to greater susceptibility to disease and other factors that cause mortality. So you have an increase in mortality rates, and that slows population growth. So what I've just described are density-dependent changes in vital rates and mortality that reflect variation in per capita food abundance. This type of density-dependent change is something that is sort of typical of top predators that are limited by their prey resources because they don't have other predators above them limiting them. And that phenomenon is what we ecologists refer to as K, or carrying capacity. Ecologists are not very good spellers, so we spell our carrying capacity with a K for some reason. So K is defined simply as the maximum or equilibrium population density that can be supported in a specific area over the long term, given the existing resources and resource dynamics. And the reason K exists is simply because the per capita availability, so for an individual otter, of their most limiting resources, which for sea otters are prey, varies as a function of population density. So you can imagine at low density, there's lots of prey. At high density, there's fewer prey. And that means resource availability affects one or more demographic rates. In this case, it's mostly juvenile survival. So that at some certain abundance, births and deaths exactly cancel out and you have zero population growth rates, so you are a K. That's all K is, there's nothing magic about it. However, K, so it's simple to describe sort of ecologically and mathematically, but how do we know when a population is at K? That turns out to be a little bit harder. That's because K is not a constant for a species. There's not some single value of K that applies to all sea otter populations everywhere. It varies as a function of habitat type, productivity, local productivity, and like Brent just described in Elkhorn Slough, there's really high productivity because it's an upwelling zone and there's nutrient input from agriculture. So that's higher productivity. Prey recruitment dynamics and a bunch of other factors. So it's logistically very difficult to directly measure prey abundance for sea otters at the appropriate scale. We can go down and do scuba dives with 1 meter quadrats, but a sea otter in a single foraging boat covers kilometers of coastline. So trying to measure prey subtidally with scuba methods over, say, a 5 kilometer square area that a sea otter might cover in 1 day of foraging is effectively impossible. So we have to use other methods. And foraging success, it turns out, the rate of energy gain that we can measure from feeding sea otters is one very useful index for assessing whether a population is nearing carrying capacity. Okay, another question that we might want to pose about carrying capacity is at what spatial scale should we be thinking about carrying capacity? Now, you may not have thought about this question, or maybe you have. We had not until relatively recently. It was simply just a question that we didn't really pose to ourselves. We thought about populations sort of at almost any scale that we happened to be studying, and we didn't really think about the scale at which K occurred. Our sort of implicit answer, at least from a management perspective, was that carrying capacity occurred at the regional scale. So for instance, we could talk about the carrying capacity of California sea otters, or Southeast Alaska sea otters, or Washington sea otters. That was sort of an implicit assumption, at least from a management perspective. It also turned out to be incorrect. And I'm going to tell you a little bit about why it was incorrect and the implications of being incorrect about that. So one of the trends that had been noticed, in fact, this is from one of Jim's papers in 1990, when he compared the rate of growth of sea otter populations in different areas, Southeast Alaska, British Columbia, Washington, and Attu are all the trends shown here, where populations that were at relatively low density were growing at about 20% per year as the growth rate, which is very close to the theoretical maximum rate of population growth that you can get in a sea otter population given their sort of survival rates and birth rates. Meanwhile, down in California, the population, which was at comparable density to these, or comparable abundance for all of California, was only growing at about 5%. And since most of California still didn't have otters, it was assumed that California population was still well below carrying capacity and should have been growing much more rapidly, similar to these populations at 20% a year. So this sort of led to this puzzle of why southern sea otters in California were growing so much more slowly. And there was a lot of research that I contributed to, as many others did, trying to sort of solve this puzzle. What was different about the California sea otters? I won't get into some of that research, although we learned a lot of interesting things conducting that research about sea otters and their health and their biology. However, before I tell you what we actually think is going on, I'm going to sort of ask you to picture in your head a simplified sea otter population and a habitat. The habitat is this white box, and we've introduced a couple sea otters here into this white box, and we're going to see what happens as they mate and make pups. So the population grows rapidly at low density. And my animation doesn't work in PowerPoint, but you can imagine these appeared sort of rapidly at first and then slowed down. And eventually they stabilize at this carrying capacity. So carrying capacity is occurring at the scale of this entire habitat or region, this white box. So this is a homogeneous, well-mixed population. Now, I want you to imagine another sea otter population, just like that. We still have our region, which is this white square, but now we've divided it into cells or habitat areas. Was it a grid of squares inside the big box? So we're going to just say that this represents spatial structure within the population. It might be different areas of coastline or different islands, or it might be nothing that we can see at all. It might be a sort of cryptic structure. So we have ask our population to grow, which it does. And it looks more, if you ignore sort of the sort of faint gray grid lines there, you might say, well, it looks like just like the previous graph. But in fact, there's different things happening here. There's localized dynamics going on. Specifically, if you look at the different habitat squares, the rate at which these uncolonized squares are colonized depends on the mobility of individual sea otters. The likelihood that this otter here is going to end up in the square next door. Recently colonized subpopulations are growing at a very high growth rate, which close to what we call Rmax. So that's about the rate of 20% per year. And meanwhile, long-established subpopulations within this area are no longer growing at all. So they're remaining at this point. So it turns out that maybe it's 6 otters per grid square or something like that. So carrying capacity, K, actually occurs at this scale, at the smaller scale, not, in fact, if we want to know K of the entire region, we have to multiply the individual Ks of all the different subsquares within there. So what I've just described is a metapopulation. And in this case, it's a metapopulation with cryptic or hidden spatial structure, but there's structure nonetheless. And that structure is imposed because the scale at which K occurs is the scale at which individual animals are essentially homogenized demographically, and at which the resource that is limiting them varies at, which in this case is their localized prey, invertebrates, benthic invertebrates. So are sea otters like the first example, or are they like the second example? And you can probably already guess the answer. After years of studying them and tagging them and looking at how they move and with home range size and what their diets are, we can pretty much confirm that they are very much like the right-hand example, the spatially structured population. And that structure arises from the extremely limited mobility of individual females. As I mentioned, individual females spend their entire lives in about 5 to maybe at maximum 25 kilometers of coastline, and they never leave that area. Occasionally, younger animals and males will make larger distance movements, and that's how range expansion occurs. But once they reach reproductive age, they are going to basically be in this small home range for the rest of their lives. That, and then the very fine scale spatial dynamics and variation of their limiting resource, which is their invertebrate prey, means that carrying capacity is occurring at very small spatial scales, really on the order of tens of kilometers of coastline is the scale at which we should be thinking about carrying capacity. Okay, so that's you know, that sounds like we've kind of been demographically geeking out here. You may be asking, why does this matter to the question at hand, to the recovery of sea otters in Oregon? Or why does it really matter at all beyond just someone who's really interested in spatial ecology and demographics? Well, it matters because it turns out that the spatial structure and local carrying capacity means that the rate and the pattern of recovery is highly dependent on the geography of the habitat in which sea otters are recovering. And so you can see in this picture here, there's two different habitats. One of these is the central coast of California, where we've conducted a lot of our work, from just south of San Francisco down towards Point Conception. The other is Southeast Alaska, where we've also conducted quite a few studies and have a pretty good idea on the sort of the ecology of sea otters in that area as well. And just looking at that, you can see that these two have, the orange is the habitat available for sea otters, that coastal habitat out to about 40 meters of depth. And you can see in California, it's this narrow fringe, this long one-dimensional strings right out along the coast. Whereas in Southeast Alaska, it's this complex two-dimensional structure of, formed by archipelagos of islands and inlets and fjords. So the geography of the habitats are fundamentally different in these areas. And that turns out to be really important for the rate of recovery. So to examine exactly how important that is, we developed a very simple spatial model of population growth. So the model tracks density of otters at points spread across the landscape. We incorporate sort of what we know about sea otter ecology. We have local density dependence, we have dispersal, that is the rate at which otters move from one square to the next, and that's informed by what we know of sea otter movement distances. And also range spread, so the rate at which they colonize new points. And to parameterize this model, we used all the data we have from 20 years of radio tagging studies of sea otters in California, Alaska, and other places as well. And once we developed this model, parameterized sort of by all of our data, we evaluated that model and we ran the model on two different landscapes, California and Southeast Alaska. And again, I will emphasize it's the exact same model run in both landscapes with the exact same parameter values in terms of age-specific survival and movement distances and local densities. So here is our model run on California. We introduced our otters in Big Sur, 150 otters in 1935. And then we run the model forward. So we're running it forward just based on model dynamics. We're not trying to make it in any way match what actually happened. We're just running the model forward as though we were the ones creating history. And after 75 years of our model run, this is what it predicts in terms of the distribution and density of sea otters in coastal California. And when we compare that with what we actually see in terms of the density and distribution, it's extremely similar. There's some very fine, subtle differences, but more or less the amount of coastline that they colonized and their overall density is largely the same between the model predictions and what is observed, which in a way might not surprise you because we use a lot of the data from populations here to parameterize our model. So maybe not that surprising, except that now we can run that same model in Southeast Alaska, and again, with the same parameters. Now we'll make it a little bit similar though to what actually happened in history. We have, in 1970, there were 3 different locations of otters in Southeast Alaska that actually added up to about 150 otters. So we have the same number of otters starting from these 3 nuclei in Southeast Alaska. We run our model forwards, and this is what we get after 40 years. And when we compare that to what the actual abundance and distribution looks like after 40 years, it's again very similar. Some subtle differences, but overall it's quite similar. And when we actually compare the numbers, the shaded areas here, the blue is the model predictions and model uncertainty for Southeast Alaska, and the pink or red or pink shaded area is the model predictions and uncertainty for California. And the diamonds and the circles represent survey estimates for California and Southeast Alaska respectively. So basically the model did a pretty good job of predicting the differences between Southeast Alaska and California, but those differences are not due to any differences in the sort of the biology of the animal. They're not due to any demographic differences. Age and sex specific survival rates and movement rates and everything were identical, of course, because we made them identical. The differences entirely arise from the geography of the habitats. It's the difference between expanding along a linear one-dimensional scale and expanding within this complex two-dimensional matrix. So that was really informative for us and really changed how we thought about the patterns of population recovery for sea otters in different parts of the world. Now, what does that mean for Oregon? Well, clearly the Oregon habitat is a lot more similar to California than it is to Southeast Alaska. So that means that we can expect that patterns of population growth and range expansion in Oregon are likely going to be a lot more comparable to what happened in California than they are to what happened in Southeast Alaska, which, depending on your perspective, might be a good thing or might be a bad thing, but it is just a thing. Another question we can ask is whether or not equilibrium densities are likely to be similar in Oregon as they are to California. And that also turns out to be something, a question that we can address with data and models. So all habitats are not equal for sea otters. We've talked a little bit about that already, and you've heard about that from other speakers. Some areas can support very high density sea otter populations, other areas support low density sea otter populations. So the question is, can we predict where sea otters are likely to occur at high densities in Oregon? Because that's going to be very useful for anticipating where there might likely, for instance, be conflicts between sea otters and shellfish fisheries and other activities, but also where there might be opportunities for conservation or tourism or other things. So it turns out that we can actually, we have available to us a way to try to predict this, and that is to apply a habitat-based model that we developed for predicting sea otter equilibrium densities in California, and applying that to the same habitat or similar habitats in Oregon. And this work I'm going to present to you now really represents the work and blood, sweat, and tears of Dom Cohn, who did his master's thesis with Lee Torres at OSU. And his master's dissertation is now actually, I think it's in the final process of being accepted as a manuscript to a journal, so hopefully soon will be available as a journal paper. And that is applying essentially the California model from this first paper to the Oregon habitat. So carrying capacity in sea otters. As mentioned, equilibrium densities and density dependence occurs at very small scales. And some of the things that, that we can measure to help us predict what local carrying capacity will be are things like bathymetry. So that includes both depth, but also the slope of the undersea benthos. Substrate type, and in particular, whether it's rocky and or complex habitat like on the right, or soft substrate like on the left. The degree of kelp cover, and although we'd like to measure all types of kelp, the one that we can measure most easily is surface canopy kelps, because we can measure that using aerial photographs. So percent kelp canopy, which provides both food and resting habitat for sea otters. And then also other oceanographic parameters, and the one we used as an index of invertebrate growth and recruitment is net primary productivity. And then of course estuaries versus open coast, and I won't talk too much about that because Brent covered that already, but there's obviously big differences between estuarine habitats and outer coast habitats. So here are some of the feature maps of some of those layers for a portion of the coast. This is Monterey Peninsula in California. So there we have our rocky substrate versus soft sediment habitat based on multi-beam sonar. And we have that for all of California coastline. We also have that for all of the Oregon coastline as well. Bathymetry shown here in terms of the color and kelp cover. And then we also have cumulative survey data for sea otters. So this is survey points from 1983 up to 2018. We can lay a 100-meter grid over the entire coastline, and then we can count up the number of otter observations in every single one of these grid squares. We didn't do this manually, we used GIS to do that because it would take a long time otherwise. So then we plug it all into a model. So the expected equilibrium density at any point G on our coastal grid can be estimated as a function of all those local habitat features. So I'm going to use this big capital H here to represent a vector of all those the values of all those habitat features. And if we sum that vector multiplied by a bunch of parameters alpha at an intercept, we'll get our expected log equilibrium density, K, for that grid cell. And if we add that up over all the grid cells, we get our total expected K for an area of the coast. And if we plug that K for the area of the coast into a theta logistic growth model, we can predict how the population should change over time. And we can compare that with our observed data on how the population actually did change over time. And what I've just described to you is a Bayesian model for estimating the value of a bunch of parameters given observed data. And you can look at that visually like this. If we have a bunch of parameters we want to know the value of for a model, we plug those parameters into some sort of process model. In this case, our process model predicts equilibrium densities at grid cells and adds them up over sections of coastline, and then predicts how population dynamics will change over time. And that leads to predicted annual counts by area and by grid cell, which we can compare with observed data and thereby get our values for our parameters. And so now you can make your very own Bayesian model. And this is what the model predicts. The gray bands are the actual predicted abundance in different parts of the coastline compared to the observed data, which is our survey points. And I'm showing you this just to show you that the model does a pretty good job of predicting how abundance will change over time and the densities at which populations reach equilibrium. And when we actually look at the density, so the density at carrying capacity for otters in different sections of the coast, so numbered here from 1 to 19, and you can see where those are along the coastline here, you see there's quite a bit of variation, and some areas in particular have really high densities. One of these is Elkhorn Slough, which Brent already talked to you about, and another one is Monterey Peninsula. But even within the rest of the area, you see there's actually a fair amount of variation in what equilibrium densities are. And if we plot that on the coast, you can actually create this sort of heat map of what equilibrium densities look like spatially explicit, in a spatially explicit way along the coastline. You can see, for instance, predictions around Point Conception of where there are high densities of otters, and those, not surprisingly, correspond to exactly where we do see higher densities of sea otters within the currently occupied range. But we can then also apply that model to unoccupied habitats, places that sea otters have not reached yet, but will be reaching before too long. And that allows us to basically predict where we are going to expect to see higher densities or concentrations of sea otters. So for instance, we can predict that along the southern edges of the Channel Islands, there's ideal habitat for sea otters, and that's where we're going to see higher densities of sea otters being supported over time. Now, Channel Islands already happens to be a national park, so that's a good thing because otters will have protection in those habitats. We can also apply the same model to Oregon, which is exactly what Dom did for his thesis. And here you see the results. So here's the Oregon coastline divided into north, central, and south, and you can see the predicted density of otters as a heat map with orange colors, or orange and red being high densities and blue being lower densities along these different areas. And again, not surprisingly, I mentioned that otters are sort of limited in depth, and so you can see, for one thing, all the high density areas tend to be shallower areas, so you don't tend to see high densities of otters far offshore. And then estuaries can support slightly higher densities than can similar habitats in the outer coast. But the really high density areas really occur in central and southern Oregon. And you can see these areas here that are with black lines drawn around them. These are sort of the highest densities areas. So in central Oregon, that would be around Depot Bay and just around Newport and just south of Newport. And in southern Oregon, that would be around Port Orford and just south of there and Gold Beach area, and then down right near the southern end around Brookings. So these are some of the areas where the habitat, at least to the extent that it's that these habitat relationships are going to be similar to what we've seen in California, we would predict the sort of the optimal habitats for sea otter populations. Okay, so next steps are to take these maps and then combine them with a dynamic projection model in order to actually begin to evaluate scenarios of population recovery from different areas and how that population growth and spread over time would look if otters, for instance, say, were to arrive at Port Orford or Gold Beach or Brookings, and where we might expect to see otters after 10 years, 20 years, 50 years. I should mention we've done this in other areas. We've, for instance, it has a similar model in British Columbia for Haida Gwaii. We have not done this model yet for Oregon, but that is sort of on the to-do list. And then, of course, we could then, with that output, we can combine with fisheries information and other human use of the marine environment type GIS layers to evaluate the potential timing and magnitude of their likely socioeconomic effects, and even begin to evaluate different scenarios as to where those might be minimized or how those might be minimized. So I'm going to wrap it up here. Just in conclusion, hopefully I've gotten across the idea that sea otter ecology has some very interesting, we've learned some interesting things about sea otter ecology over the last 20 to 40 years. And some of these things really help us to understand the very strong ecological interactions and their status as a keystone species, as Jim described the other night. These aspects of the ecology, though, are often quite complex. So foraging behavior and food web interactions are not fixed or sort of deterministic and identical no matter where you go. Turns out they vary quite a bit by habitat, but also by population density and even within individuals within a population. And those complexities have real world consequences in terms of how these food web dynamics play out in different times or in different places, and as you get different perturbations to the food webs. Spatial structure and local density dependence, those are some big sounding phrases, and I could probably have phrased them better, but the fact that sea otters are limited by their prey at relatively small spatial scales of the coast turns out to have really important consequences for how their populations change over space and time, how they grow and recover in different areas. And that, in particular, the geography of the habitat turns out to really have a strong determination on the rate and patterns of population recovery. and we believe that Oregon, being more similar to CA to California, is probably going to exhibit more similar patterns to California. And finally, hopefully I've convinced you that understanding sea otter foraging ecology and population biology can be a useful tool for helping us to anticipate some of the patterns and timing of those ecosystem effects and socioeconomic impacts in Oregon over time. With that, I'd like to say thank you to a lot of people that contributed to this research. There's a small and partial list here, and of course, funding agencies. The sort of patterns I talked to you about today were really the result of many, many different studies over many different years. And here are some of the agencies you see in the bottom that have contributed to that. So thank you very much, and I'd be happy to take any questions.
Well, thank you very much, Tim. And although you can't hear it, there's a lot of applause behind the virtual silence. And I will go to the first question. Okay, about diet studies at the different study locations, did you measure the quote-unquote availability of the various prey species consumed by the otters and determine the relative consumption rate of those prey?
Yeah, so the answer is kind of to that is sort of a yes and no. That comparative study I showed you for San Simeon, the Cambria area, and San Nicolas Island, we did in that case, at least for the prey, that there are methods that we can measure those prey. So there happen to have been long-term scuba-based subtidal survey programs in place at both of those areas, which I thought was actually one of the factors that contributed to our decision to use those two areas for comparisons. So we actually did have really good information for those two areas for some of the more important species in terms of what their benthic availability was. And that, so yes, in that case, but it's a highly qualified yes. And that's highly qualified yes, because some of the prey types, there are no reliable ways to measure underwater, or they're very imperfect. For instance, crabs is a good example. We can use catch per unit effort, which we have, and that's what we used actually for studies in Elkhorn Slough as an index of relative abundance. But any scuba-based methods that we have for measuring crab abundance really are not very adequate for measuring them in the way that sea otters encounter them. The other major problem you have with that is, again, that issue I mentioned of spatial scale. Sea otters forage over a they have small home ranges, but an individual foraging bout can cover a large portion of their home range. So 1 2 to 3 hour foraging bout might cover 3 kilometers of coastline. And we don't really have measurements of availability at that scale. So that does complicate that ability, sort of the way that we typically measure use versus availability. We've gotten around it in several ways, and again, by using scuba data, scuba-based survey data as much as we can. I don't know if I answered the question very well, but it's so yes and no.
Yeah, thank you. And I've got 2 questions here that are kind of related, so I'm just going to read them one after another. If an individual otter is already an urchin specialist, does the nutrition derived from urchins decrease after an urchin boom, or zombie urchins, quote unquote? And really the related question from James is, did urchins in Monterey raise the energy intake rate of either specialists or other diets post-urchin bloom?
Yeah, great question. So, so first off, yeah, and assuming you might have seen Josh's talk the other night, one of the things we found with our urchin specialists is that they were very good at finding where the energy-rich urchins were. And so Josh showed you diets or observations or maps of where these urchin feeders were capturing urchins and consuming them, and those tended to be really in the kelp, both the patches of kelp that were not in urchin barrens, or right along the edges of the urchin barrens, and often just like maybe 10, 15 meters from the edge of an urchin barren. That's where we would find our otters feeding. Many of those dots that he showed in the map, actually a majority of those dots that he showed in the map were actually from our tagged otters. So we actually knew what their diet specialization was. And most of those otters catching urchins, of course, not surprisingly, happen to be urchin specialists. So the urchin specialists are really good at figuring out where the productive areas are, and those are the areas that they tend to focus on their foraging effort. And then that's to the second part. Yes, we did see the overall rate of energy gain increased considerably, actually, in Monterey. And it turns out it wasn't just urchins. We focused on urchins, or I focused on urchins today, again, largely for consistency with the Josh's talk, and because we've been particularly interested in sort of that effect. But another species that really exploded in abundance after 2014 were mussels. And so the 2 diet specializations that that more than doubled in frequency within the population were urchin specialists and mussel specialists. So it turned out before the 2014 urchin boom, well, urchin boom and mussel boom, urchin specialists and mussel specialists were relatively small portions of our study animals, and had been really for the previous 15 years that we've been conducting studies. But after that time, during our 2015 to 17 study, or actually that study's really just wrapped up in the last year, but a really high proportion of our otters were either turned out to be mussel specialists or urchin specialists. And most of those were younger animals. So again, they came up in that world of really abundant urchins and mussels. And they had very high rates of energy gain, at least over the first years of the study. We're starting to see that rate of energy gain and the population, increase in population abundance of otters around Monterey begin to level off and actually decline again. So sort of the heydays of that, of the bonanza of abundant food seem to be tapering off. It was really from 2015 to 2018 were kind of the boom years.
Thank you. This is the next question from Steve. And Pierce and Hines, 1987, MEPS described a purple urchin boom in the 75, 76 in Monterey Bay. So it seems like the urchin boom happened, albeit at a smaller scale, while otters were present.
Well, yeah, although otters were just in sort of the recovery stage at that point, and still had more abundant other prey, including red urchins. So yeah, you're right. There was that boom then, but it was that otter population was at a very different status than it is now. But that's a great point.
A question from Walter. What do you think the future, what do you think the future holds for this new young cohort of urchin specialist otters? Say if urchins return to normal densities, i.e., will they switch, starve, and might this provide increased control of urchins?
Yeah, that's a great question. And I think we're going to have, we don't really, since we've never seen anything exactly like this before, we're going to just have to keep watching. Although sadly, that study is, at least the otter portion of the study is finished. And we're no longer able to keep tabs on those otters anymore, or at least at the same level. Some of them are still tagged, and there are still some data coming in, but it's largely tapered off. But in terms of, at the population level from surveys, we should still be able to keep tabs on what's happening numerically with otters. It's going to be really interesting to see. We have seen, there are some other incidences of sea otter populations responding to episodically really abundant prey and then switching back again when that prey disappears. The most dramatic of those are the explosion of lumpsucker. Is a really bizarre fish that occurs in the North Pacific, and it's a pelagic species that has this largely unpredictable nearshore spawning events that occur periodically, but in different places. And in the early 90s, there were these huge spawning events of lumpsuckers in the nearshore, both at two of the islands that we conducted studies, Amchitka Island and Adak Island, and that occurs in the spring. And it just so happens that those spawning events occur right when urchins have just spawned out and are very low nutritional value. And typically those are the times when we see the highest otter mortality. But for a couple of years there, we had these super abundant lumpsuckers, and all the otters switched to lumpsuckers. And it was almost, and from our foraging data, it really shows up. We have all these other prey that suddenly drop out, and every foraging bout was an otter eating a whole bunch of lumpsuckers. And that led to really, really low mortality rates during those years as well, because they had this super abundant prey. But then once the lumpsuckers disappeared, they went back to their previous diet specialization. So I think that's sort of one example, and there's pelagic red crabs was another that happened in California. So I think it is likely that they will switch once that sort of bonanza of prey disappears. How it disappears, or if it disappears, of course, is another question. If an urchin disease, for instance, swept through the population and sort of decimated urchin populations, then they'd probably have to switch very quickly. So yeah, I think time will tell how things play out.
Thanks. Next question on a related topic. Marcus asked, would an otter keep its food specialty for a certain length of time if it were relocated, or would it change? And maybe a related way to rephrase that is would you recommend or consider any sort of strategy with respect to what otters might be the best relocation candidates, one way or the other?
Yeah, that's a really interesting one, and Lillian Carswell, who's may or may not be listening in, might have some thoughts on this, because she did her master's on that very topic, thinking about what were sort of the pros and cons of different ages of otters for relocation or for introductions in a translocation event. I think I see Lillian is there, so I'm not sure if she can.
Just moved her over to be a panelist. So.
Okay, yeah, I'll maybe Lillian may or may not want to chime in here, but I don't want to put her on the spot. But it's one of the factors we think that contributed to a lot of the original otters that were translocated to San Nicolas leaving and going back. And with hindsight, when that translocation happened, not a lot was known. Actually, very little or nothing was known about individual prey specialization in otters. But a lot of those older females that were translocated out swam hundreds of kilometers back to their original home ranges from San Nicolas Island. And with hindsight, that's probably because we put animals in a place where they were exposed to different habitats and different prey than what they were used to eating. And interestingly, at that translocation site, during the first years after the translocation, some of the otters that did stay were eating had quite a diverse diet, but after about 3 or 4 years, they all began to switch to eating the most energetically abundant or profitable prey there, which was large red urchins. So it seems that there could be a bit of a, I guess, a lag time in prey switching caused by the individual specialization. And that might suggest that younger animals, which are still able to sort of, or still have more plastic diets, might have some advantages. But of course, those animals also have lower survival rates. So how that trade-off plays out, I'm at this point, I don't have any strong feelings, and I'm not sure if Lillian does. Lillian, have I botched that, or do you want to say anything about that?
Hi, Tim, thanks. I'm not sure if you can hear me.
We can hear you.
Okay, great. So I don't have much more to offer than Tim already said. I wasn't looking at diet specializations, but I was looking at the rates of departure of animals from San Nicolas Island. And so these were all wild translocated sea otters, and they're known to have pretty strong site fidelity to their home range. And so a great proportion of them returned exactly to their home range. And I didn't look into the causes of that necessarily, but I think it's certainly plausible that you know, they found themselves in a habitat type that was unfamiliar and with prey types that were available at different abundances that they were used to. But yeah, I think there's a lot that we still need to understand about why sea otters home and what it is about their home range that that is significant to them. I mean, obviously, food availability is one of the most significant things for an otter, but you know, how they're sensing their environment and what they're using to cue in on, you know, what is my home range and how do I get back there are still really interesting questions to explore. And so the very strong homing tendency of sea otters, of wild sea otters, is one of the reasons that we've been talking so much more about using rehabilitated stranded pups that have been raised in a tank and they're young and they really don't have a wild home anymore. You know, they don't remember the beach they stranded on at two weeks old. And so that is a potential way around the really big losses that we see when wild sea otters are translocated.
Thanks, Lillian. Okay, I'm going to try to give a couple more questions here to Tim, and maybe we'll fit in a few for Brent also. One from Larry says, curious why models for Oregon does not predict higher otter populations in the vicinity of Cape Arago, Charleston, where there is at present abundant habitat and at least historically sizable prey base.
Yeah, that's great. Well, you know, that's a great question, and I guess that's a good reminder that that all models are wrong, but some are useful. Yeah. Most famous phrases ever used, and every good professor that teaches modeling opens with that phrase. It's so the model is a gross simplification of reality, and B is limited by the data that we feed it. So what we would really love to know if you're going to model habitat for sea otters is the availability of all the different prey species in at every different grid cell. If we knew that, we could probably predict largely almost, well, we'd have to add, you know, pupping habitat and things like that, but we could probably almost predict perfectly what the abundance of otters would be if we knew their abundance of prey and the productivity of the prey or the recruitment rates. And of course, we don't know that for almost any of their species. So all the features that I used in that California model were things that we could measure that we thought would reasonably be predictive of or related to what the things that actually matter for sea otters, which is the relative availability of preferred prey. And that, so they're really indexes for that. And they did they did a very good job of predicting areas of high and low density within California. But one then has to remember that the things that predict good prey for otters in Oregon may be slightly different. In fact, they may be different, quite different even. And so the model it wouldn't surprise me at all that the model would end up in some cases predicting high densities in areas where there aren't good prey, and vice versa. And that is just a limitation of models. They are potentially very useful tools, but they are just tools, and they're definitely not perfect. So I guess that's may sound like a cop-out, but I think finding areas where models aren't doing a good job is actually one of the most interesting things we could do. So I'd be definitely interested to talk to you more about that and find out, and get a sense for why the model might not be performing well in that area, and we could maybe find a different data layer to plug into it.
Thanks. And just maybe one or two more. I've got kind of two tandem questions. One from Madonna is, given the large sandy coastline or large stretches of sandy coastline that occurs between rocky shoreline stretches, is there therefore an implication of your research that the Oregon sea otters would have to be reintroduced at multiple locations?
So you might have noticed that big difference in the growth rate. Some of the difference in that Southeast Alaska versus California comparison, most of the difference was caused by the different topology of the coastline, but also some of it was the nature of the fact that there were three locations there. And actually for all of Southeast, there were 7 different locations that otters were released at. And so the people that did that made those decisions, I think, had a very strong intuitive sense of what was going to work really well in terms of maximizing the rate of spread, or maybe it was just total luck, but they couldn't have done a better job at maximizing the potential growth rate in Southeast Alaska, given even if they were the most amazing spatial modelers of their day. And that's just not only where they the sort of spatial spread at which those release points occurred. And if you were to model the Oregon population or the California population from like 2 or 3 different nodes spread out along the coast versus just one, you get really different results. Now that's assuming that each of those nodes stays where they're put, and otters are known not to always stay where they're put. The past Oregon translocation is a great example. So you could put them in 3 or 4 different locations, and they might all consolidate into one location, or conceivably they might all swim up to Washington as well. It's hard to predict what otters will do, but with that caveat in mind, that otters don't necessarily stay where you put them, it is definitely a fact that you will get a much faster rate of recovery or overall if there are multiple nodes from which the population is spreading from. That may or may not be a desirable thing, given sort of the various pros and cons of sort of the rate of recovery. But yes, it's definitely a consideration.
Thank you. And I just throw one last one at you so we don't drag you into a super long Q&A. But one question from Scott is, thanks, Tim. Can you estimate or speculate on the total abundance of or total population of sea otters that could be supported along the coast of Oregon?
It's actually in Dom's thesis and in his paper. Well, the paper I think we might have updated from the thesis by running the final version of the model. I actually don't have that number right handy. It is I can definitely find it, and it's to say it will be coming out hopefully fairly soon in the paper. It's in the 5,000 general range, but I don't want to say a specific number because I don't have it handy. So yeah, there is a number that comes from that model, and it is subject to, and there's a wide confidence limits around it, very wide confidence limits around it because of all the uncertainties involved and the fact that it is a projection based on habitat relationships from California that have been extrapolated to Oregon. So in the paper, we're pretty cautious in, in that specific number.
Well, thank you, Tim, and I'm going to just allow a couple more questions for Brent, and I want to just apologize to some of the other folks who had a lot of questions coming in, but we will try to accommodate maybe and relay those questions to some of the presenters by email.
Oh yeah, sure, that'll be fine.
For Brent, we have one question from Liz, and what do we expect the impacts to be for mariculture of oysters and clams in estuaries?
Yeah, so that's a really important question for the clams. You know, we're in Southeast Alaska, on the team I'm working with up there, the Apex team, we call it. We're looking at that right now, and in places like Southeast Alaska, it seems that predation on clams, especially, you know, the big threat is clam gardens from indigenous community clam gardens are under big threat. So it definitely poses a risk. The oyster question is very important. It's even in California, because, you know, Tomales Bay is one of those estuaries near where we're kind of targeting, and there's significant amounts of oyster mariculture happening right there. And so we've been in contact with those folks to kind of explore, well, would there be an impact? Can even sea otters eat oysters? There's some evidence from Alaska that they can, but it's really going to depend on the technology that the oyster farmers are using to grow their oysters. You know, some use flimsy, plastic bags hung on PVC poles. Some use metal cages, which would probably be the ideal way to go if sea otters were to be relocated, reintroduced, or recover. But it's an unanswered question, and I think, if sea otter expansion is going to occur, I think conversations and maybe even research in partnership with oyster farmers and even maybe clam farmers is going to be really, really important, because right now with the clams we kind of understand that there could be impacts. The oysters we just don't know.
Gotcha, thank you. And this question is from Stephanie, and she asked, do you have any examples of the type of management that would need to be done in order to have sea otters in San Francisco Bay? And I'm going to kind of expand that question to include, and it doesn't have to be site specific, but thoughts that you may have and some of the other panelists may have about management in bays in Oregon and in general, so to help accommodate a reintroduction of sea otters into those type of habitats.
Right. Well, I think, the first step, there's got to be some sort of community buy-in. I think if the community is not receptive of sea otters, then there might be some problems down the road. So I think community engagement is super key early on. It kind of goes beyond some of the science. But, having managers, fishermen, NGOs, scientists kind of all on board on the same page is going to be kind of ideal to making those types of decisions and trying to predict, what's going to happen to some degree. I know Tim's been downplaying his models a little bit. Tim, I wish you want to do that. I mean, I think the models for that aspect are going to be almost essential for trying to predict what could happen. And that's getting an assessment of the state of the ecosystem and the habitats, kind of like we did in Elkhorn Slough. But yeah, also kind of thinking about all the local factors, the societal factors that would be at play too. And then, the political aspects of it as well. There's, permits are needed, and, things like, the Endangered Species Act and Marine Mammal Protection Act also probably need to be considered quite intensively before making those kinds of decisions.
Well, thank you, Brent. Well, I think I'm going to hand it over to Bob, if maybe he has a wrap-up question or any thoughts. But and we're going to try to relay, if the panelists are okay, provide an opportunity for some of the folks listening to maybe shoot you an email with a follow-up question if they didn't get it answered, if that's okay.
Absolutely.
Absolutely.
Great. Okay. Well, let's a big round of applause out here in cyberspace around the world for Tim and for Brent and really all of our speakers and participants. This has just been fabulous to be able to sit and share and learn this research and to really get a peek at the issues and the science that's going to be needed to be addressed as we go forward in considering bringing sea otters back to Oregon. This proves that it's almost always the case that when you don't know anything about something, things seem simple. But the more you get into it, the more complex it becomes. And that's what we're learning here. And I really appreciate, we on the board of the Elakha Alliance, appreciate the time and the expertise and the passion with which various of our presenters today and their colleagues really approach these issues, and their willingness to contribute to our effort by sharing their knowledge and the fruits of their work. We will do our best to follow up this symposium with links on our website to as many of the relevant papers as we can. Certainly Dominique's master's thesis about reintroducing sea otters to Oregon is a key one. I think that's already on our website, linked to our website. So we'll do that. We'll be sending you a follow-up a link so you can do a little survey about this thing. And I would encourage you to stay tuned and go to our website, be on our mailing list, get our RAFT newsletter, and keep up with what we're trying to do. This is not going to happen overnight, but as we make progress, we can see that there's the possibility of bringing these really fabulous little animals back to what was once their home, and repatriate them to the ecosystem with the benefits that go with it, both in the nearshore environment and the estuarine environment. So that's our goal, and we'll see how it plays out. Thank you, everybody, for attending, and have a good afternoon and evening.
Thanks, guys.
Thank you.
Thank
Bye.
you very much.
Yeah, thanks.